Mhm. So this is a constant link for the lecture, and you will take 8 lectures during this term, once at 2 weeks, and you should enter the lecture by using of this link. The leader should inform the students. The first lecture is, the first lecture is the subject of microbiology, morphology, and systematics of bacteria. Microbiology, you will study microbiology. Microbiology is the study of microbes. Microbes are particles that are visible by using of microscope only. Microbes did appear on the Earth many billion centuries ago, and microbes have a tremendous impact on all life and the physical and chemical makeup of our planet. They are responsible for cycling the chemical elements essential for the life. And microbes did create the life in nature, natural life, like now we see this natural life. And microbes are very adaptable. It's possible to detect microbes any place on the planet, like in the soil, on the bottom of ocean, in the hot water, in the cold water, in the air. And when animals and human beings did appear on the earth millions centuries ago, some microbes enter animals' body and humans' body, microorganisms' body and plants, and they did decide that there are good conditions for them. And I hope you know that human's body, even the person is healthy, absolutely is not completely sterile. All organs and tissues of human's body, those are connected with environment, have microbes. And the microbes enter human's body immediately after the newborn appears. Fetus inside the mother's body is sterile if mother is healthy, is not infected by some microbes. The fetus is sterile, but immediately when a newborn will breathe the air and in contact with some hands of medical staff and so on, and due to feeding, the organism of newborn will get microbes. Of course, the only internal organs, those are inside the body, and the internal liquids are sterile, they still sterile. Other tissues like skin, like mucosa of eyes, mucosa of nose, mucosa of throat, mucosa of genital tract, and all complete digestive tract, of course, they have microbes. Microbes are recognized by immune system of human's body, and multiplication of them is suppressed by immune system. They are not able to multiply they wish. And multiplication of them is constantly suppressed by immune system, and they develop specific biotopes in every organ, in the nose, in the mouth, in stomach, and there are very complicated relationships in between microbes, and some of them suppress multiplication of another, like bacteria suppress multiplication of fungi. Some fungi, like Candida, genus fungi, are detected in some healthy people on mucosa of mouth, mucosa of genital tract. In normal condition, they are detected just a few cells visible in microscope. So, but if the immune system is a little bit weak and a temporary immunodeficiency is developed, due to maybe high temperature, low temperature, immune system is very sensitive, maybe overdoing stresses and some somatic diseases. If immune system is a little bit suppressed on the background of immunodeficiency, these microbes named normal microbial flora, they will get opportunity to multiply, and they will multiply, and they are able to cause infectious diseases. So, by this reason, all representatives of normal microbial flora are named conditional pathogenic, and infections caused by them are named opportunistic infection, from the word opportunity. You will study normal microbial flora later, and you will know that they play a positive role. And on one hand, and on another hand, all of them are able to cause endogenous opportunistic infections without any contamination from outside. So, normal microbial flora, microbes are able to cause infections. And at the same time, most microbes still survive in nature, in the soil, in the water, and they don't need human's body, animal's body. They are named saprophytes, but occasionally they enter human body, like by inhalation of spores of fungi. There are many species of fungi molds. Those exist in the soil, and there are favorable conditions for them. But occasionally, by inhalation of particles of dust with spores of these fungi, people are infected by fungi, and they are able to cause very severe diseases named systemic mycosis, disease caused by fungi and mycosis. Bacteria, Legionella genus, exist in natural water, and they don't need animal's body or human body. They enter human's body by inhalation of droplets of natural water, and they are able to cause legionellosis with clinical symptoms of fever and pneumonia, and sometimes they lead to lethal outcome. So, and at the same time, microbes are able to infect human beings and cause infectious diseases, sometimes with lethal outcome. So there are pathogenic microbes. Pathogenicity is ability to cause infection. So between people, and some microbes are transmitted from animals, there are microbes, those have specific factors of pathogenicity. They are able to cause the dysfunction of many organs in human body, and sometimes it leads to lethal outcome, means death of the patient. Such microbes are named pathogenic microbes. So you will study just one branch of microbiology named medical microbiology. So you are not going to study saprophytes. A lot of microbes, those exist in nature, in the soil, in the water. You will study just microbes, causative agents of infectious diseases, pathogenic, conditional pathogenic, and a few species of saprophytes. And from the ancient centuries, scientists did develop ideas about microbial origin of some diseases. They did notice that some particles are transmitted between people. The first one was Hippocrates has entered into the practice the miasmatic theory of development of diseases, connecting their occurrence with inhalation of certain harmful evaporations. He gave the name miasmas, Hippocrates. He did notice that some particles are transmitted from the patient to healthy people. Then another one, Titus Lucretius Caro, has assumed presence of the certain pathogenic seeds getting in an organism of the humans and animals, causing development of diseases. Next one, more precisely, the nature of infectious diseases has explained by Girolamo Fracastora. He did live in the middle centuries, and he considered that infectious illness transfers certain alive pathogenic agent, contagium vivum fluidum in Latin, and he has specified materiality of this agent, and he's entered the term infection. Girolamo Fracastora, very famous doctor, and the first who managed to penetrate into the world of microbes was Anthony van Leeuwenhoek, creating microscopes. He was just producer of lens, and he did examine some drops of water and other substances by using of his lens. And he did notice some living particles that did move in the raining water. And so he is obliged to him opening of microbes. He did describe animalcules. He did notice some living particles that did move in raining water. And he did describe report. He did send report to scientific society. And it was the first fact when some living tiny organisms were detected by using of microscopes. And then after that, this idea were developed, and it was the beginning of microbiology. Finally, scientists, doctor did realize that microbes are able to enter human body. They are able to multiply in human body and cause infectious diseases. And one by one, many causative agents were detected. Bacteria, protozoa, fungi, viruses were detected much more later. People did die of infectious disease at that time. The doctors had no any preparations for treatment. They did use some toxic substances, tried to kill microbes. Of course, it was not a good idea. And at that time, they had no any measures to prevent infectious disease prophylaxis. And Edward Jenner, the English doctor, has proved that the infection with cowpox virus is effective for prevention of a natural smallpox. That is common for people. At that time, there were epidemics of smallpox, and many millions of people did die of smallpox. And in 1796, he has inoculated to the 8 years James Phipps the fluid from the pustules of the milkmaid Saranaems. He did notice that women that did take care of cows were infected from cows by cowpox virus. Cowpox virus is not dangerous for human beings, but by infection of this virus, some pustules were developed on the skin. And he did notice that this woman finally were not infected by smallpox virus. He didn't know that it is virus. He did realize that some microbes are transmitted. And he did this attempt. He takes some specimen from the pustule of milkmaid, this woman who did take care of cows, and did inject into the skin of this boy. And so he didn't develop theory of vaccination, but really it was the first time in medicine when the person was vaccinated. And he did protect many people by this procedure, but he did not describe the scientific theory. He was a doctor. He was very busy, but he did it, and he did save the life during the following of 2 years. He was inoculated more than 100,000 persons, and he did help them. The first scientist who did develop the theory of vaccination was Louis Pasteur, French scientist. Louis Pasteur really was a chemist by occupation. He was not a microbiologist. And some producers of wine in France, they did ask for him to help them. They did notice that the wine is spoiled, and they did ask for him to realize what chemical reactions are going on in this wine. And Louis Pasteur did detect bacteria, and he did suggest them to heat this wine for a short time very quickly, so and to kill them, to kill bacteria. And so after his name, Pasteur, now pasteurization is very widely used in our life. Milk, juices, beer, and other due pasteurization are kept now for a few days or weeks. Pasteurization now is very widely used. After this fact, he was interested in microbiology, thank you goodness, and he became a microbiologist. He will study some microbes, some causative agents of hence, and he did notice that it is possible to decrease the virulence or this pathogenicity. He did notice occasionally but he was a scientist, and he was thinking about it, that it is possible to reduce the virulent properties. Pathogenicity, the property I'm repeating, pathogenicity is ability of species of microbes to cause infectious diseases. They have special factors of pathogenicity. And inside the species, strains are different by these properties and virulence. Every strain of microbes has ability to cause infectious disease. So virulence and pathogenicity are similar terms. Pathogenicity is ability of species of microbes to cause certain infectious disease like Vibrio cholera bacteria. Species Vibrio cholera bacteria are able to cause exactly cholera. Corynebacterium diphtheria species bacteria are able to cause exactly diphtheria. Mycobacterium tuberculosis bacteria are able to cause exactly tuberculosis. So, pathogenicity is the ability of species. Influenza viruses are able to cause exactly influenza. Do you understand me? But in abstract, I'm talking about species of microbes in abstract, but in real life, there are real strains that are circulated between people or animals. Strains and ability of certain strain to cause diseases is differ. And virulence is the ability to cause infectious disease of certain strains of Vibrio cholera, of Mycobacterium tuberculosis. They're similar, okay? So, and he did note that it is possible to reduce this ability to cause diseases. And from this fact, the attenuation theory has developed. This reduction of virulent properties is named attenuation. And he did develop attenuated strains that did not cause the infectious diseases, but finally they did develop immunity to infectious diseases. So the first artificial vaccine has appeared. It was vaccine against anthrax. Again, it was used for animals. Again, French businessmen did ask for Pasteur to help them and to save cows. At that time, anthrax was very common in all countries, and animals and human beings, those who were infected from animals, did die. Thousands of animals and many thousands of people every year did die of anthrax. So, and Louis Pasteur did develop vaccine for some preparation for specific prophylaxis of anthrax in memory of the Jenner, Edward Jenner, who did it first time. He has named the weakened attenuated microbes as a vaccines because of Vexa. Because of Vexa was a disease, and safety inoculations was named vaccination. Then Louis Pasteur, he didn't know viruses. Virology was not developed at that time. He did develop a vaccine for specific prophylaxis of rabies. He did note that he did realize that rabies is transmitted by some microbes, but he didn't detect these microbes by using of light microscope. It was not possible to see viruses by light microscope. Electron microscope was not developed at that time, and virology was not developed. But he did note that it is possible to cultivate these unknown microbes in the brain of laboratory animals. And he took the brain of dog with rabies and did inoculate the brain into the brain of laboratory animal rabbits. He did cultivate, and then he took this brain and he did use high temperature, low temperature, some chemical, and finally he did decrease the virulence of the rabies, the virus, and it's still alive. Microbes still alive, but they had the low virulence. They were not able to cause severe diseases, just very mild form. And he did use this vaccine for animals. And finally, he did use one time for one boy. He was afraid to use for human beings, but he did it. And from that time, vaccination from rabies was started at different countries by use of his vaccine, Louis Pasteur. After that, his colleagues, Calmette and Guerin, did develop vaccine for specific prophylaxis of tuberculosis that is used till nowadays in all countries, named BCG, Calmette and Guerin bacillus. Bacillus of Calmette and Guerin, BCG. After that, many different vaccines were developed now all over the world, and many millions of people still alive due to vaccination, immunization. Okay, then Robert Koch, father of bacteriology, German scientist, introduced staining technique. He did study many bacteria. He did isolate Vibrio cholera. He did isolate tuberculosis bacteria. And he introduced staining techniques for bacteria and also methods of obtaining bacteria in pure culture using solid media. And he discovered causative agents of tuberculosis and cholera. He was very famous at that time. And virology. The history of virology begins on February 1892, when Russian scientist Ivanovsky has informed in the Academy of Sciences about opening of the activator of a tobacco mosaic, plants diseases, which he was made at Odessa Bacteriological Station. He did describe viruses that he did open in plants, and then other viruses were detected. Then Ilya Mechnikov, Russian scientist, discovered the phenomenon of phagocytosis and elaborated the phagocytory theory of immunity. Later, he did get the Nobel Prize. And he was director of Institute of Pasteur in Paris. He did work in France, finally. So Ilya Mechnikov did discover the phagocytory theory of immunity, cellular immunity. He did develop cellular theory of immunity. At the same time, at the beginning of last century, another one scientist, Ehrlich, did develop some proteins in the serum of blood of patients that did recover of infectious diseases and did develop the humoral theory of immunity. Mechnikov, cellular theory of immunity, he did detect macrophages that did take the microbes, and due phagocytosis, they did kill them, destroy them completely. At the same time, particles were detected by Ehrlich, proteins named antibodies or immunoglobulins, and Ehrlich did prove that exactly these proteins are able to connect with microbes and develop complex antigen antibodies, and this complex is eliminated. It is taken by macrophages and they are eliminated from human body. Both scientists were right, and both of them did get Nobel Prize, finally. Until now, we know that there are two kinds of immunity in human body, cellular immunity and humoral immunity, mediated by immunoglobulins. Both of them are specific factors of immunity in human body. So this is introduction, history of microbiology. And the knowledge of microbiology is very important for all people. Nowadays, I guess all mankind did realize that microbes exist and we live in microbes world. And some people don't believe in microbes till now. They don't believe, but I'm answering, don't believe in microbes. Microbes believe in you, okay? And microbes enter human body without any invitation, and they do what they wish to do, and they kill people. Did you notice tiny viruses, not visible absolutely? They're circulated between people, and people die one by one. And really, we live in microbes world. They are hosts on the earth, and we try to do something. We are try to protect ourselves from microbes. We did develop antibiotics. Microbes did develop resistance to antibiotics. Then you will study how it is possible for microbes to develop resistance, natural, acquired, and other. And it is a war really between human beings and microbes. And we are not winner in this war. Did you notice? Okay, so, and it's very important for medical university students, of course, for pharmaceutical faculty. Of course, you have to know all preparations, those are produced for treatment of infectious diseases, all preparations also produced for specific prophylaxis. You should know better than other students. Even you're not going to finish to graduate medical university, maybe. Anyway, this knowledge is very important for everyone. All people have to know where microbes exist in nature, pathogenic microbes and conditional pathogenic, how they enter human body, what transmission ways, what they do in human body, what clinical symptoms are dangerous, and you should visit a doctor immediately, and what methods are used for proving of diagnosis, preparations for treatment, and how it is possible to prevent yourself from microbes. There are two kinds of prophylaxis or prevention. Specific prophylaxis, of course, vaccination. Fortunately, many vaccines are developed, and even vaccine is not developed, non-specific prevention exists, of course. It depends on transmission ways, maybe boiled water, maybe wash hands, maybe to kill cockroaches, maybe protect from the biting of mosquitoes and ticks, and maybe avoid contact with homeless dogs, and so on. And you will get this knowledge on microbiology. And believe me, it is life important knowledge. So microbiology is very important subject, okay? Now, you will study medical microbiology. Is a study of microbes, quantitative agents of infectious disease of human beings. Pay attention, we are not going to study all microbes, those exist in the world. Maybe you remember from biology, algae. There are microscopical algae, but they don't cause infectious diseases, and medical microbiology is not going to study, okay? You will, what? Microbes is common term. Microbes are microorganisms, they are synonyms. Microbes is better because of not all of them are organisms. So microbes, common term, it includes prions, viruses, bacteria, fungi, and protozoa. You should learn by heart like 1, 2, 3, 4, 5, okay? So prions are just protein particles, just proteins. They have no any nucleic acid, but nevertheless, a disease caused by prions are slow infectious diseases. People are infected from animals and by other reason. And prions cause disease in animals and in human beings. And in spite of they are not able to reproduce, they are not able to multiply, finally, they are able to destroy the head brain completely, and it is looking like sponge, and it leads to death. Till now, no treatment of disease caused by prions in medicine. And the diseases in human beings are named Kuru, were detected first time on New Guinea Islands, then in European countries and other countries. Creutzfeldt-Jakob disease was detected, and then Gerson-Streusel-Schenker disease was developed, and then fatal familial insomnia. Unfortunately, we know more and more disease caused by prions. You will study prions later, autumn term. You will realize how are they transmitted and pathogenesis. Viruses cause viral infections, and very many infections really caused by viruses: influenza, measles, mumps, rabies, poliomyelitis, COVID-19, and so on. Bacteria cause bacterial infections. Prions and viruses have no cell structure. They are acellular organisms. Viruses belong to kingdom Vira. Bacteria, fungi, and protozoa have cellular structure, but bacteria are the most simplest cells. They are prokaryotic cells. They have no any internal membranes inside. And fungi and protozoa are eukaryotic cells. They have all internal membranes, including nucleic membranes, so they have real nucleus. And protozoa have a few nucleuses. And bacteria and fungi belong to plants kingdom. Protozoa belong to protozoa kingdom specific. So acellular, prions, viruses, and cellular: bacteria, fungi, and protozoa. Bacteria prokaryotes, fungi and protozoa eukaryotic cells. When all these microbes are able to cause infectious disease. Prions are an exception. Incubation period is long and duration of disease. So I'm talking about viral infections, bacterial, mycosis, and protozoal. As you know from your life experience, I hope the beginning of infectious disease very often is acute. Like yesterday, everything was okay with person, and today he did realize, he or she did, the person did realize that something wrong. He is not feeling well, headache, sore throat, maybe pains in abdomen, and this person will visit a doctor, and doctor will write the complaints of patient, do physical examination of patient, and give questions about doctor knows transmission ways of microbes, and doctor will ask, well, maybe he was bitten by some insect, maybe he did eat the canned food, maybe he was in contact with some homeless dogs, and so on. And doctor will write diagnosis of infectious diseases. It is clinical diagnosis anyway. The doctor has idea that some microbes are in person's body, specific causative agent. Every infectious disease is caused by specific causative agent. So the diagnosis should be proved, and it should be proved by microbiological diagnosis. And 6 methods are used all over the world of microbiological diagnosis. And there are instructions for microbiological diagnosis of every infectious diseases. And it is written what method should be used, what specimen should be taken from patient body and sent to the laboratory. There are specialized laboratories, bacteriological laboratory, virological, mycological for detection of fungi, protozoological, serological, and molecular genetic laboratories. And doctor, sometimes 6 methods are used, all of them. Sometimes 5, 4, 3, depends, at least 2. By any method, we should prove the diagnosis. We should find microbes, or we should find antibodies by using of serological method for microbes, or maybe we have to detect allergic reaction. What methods? Microscopical method, bacterioscopical, viroscopical. We try to see microbes by using of microscope. It's not the main method because not possible to realize what species do these microbes belong to. Detection of species name is identification. Not possible to identify microbes by microscopical method, but it is used like first step. Then golden standard for many decades was culture isolation. Bacteriological, virological, mycological, protozoological, isolation of pure culture, and by detection of properties, identification. It was the golden standard. Now modern methods are used very widely, like molecular genetical method. Anyway, it's still the main method. Biological method by using of animals. Sometimes it is necessary to use laboratory animals for bacteria, microbes isolation, or maybe toxin detection. Serological method is used very widely now. Again, the aim of this method is detection of antigens of microbes, or maybe antibodies for microbes in the serum of blood by using of immune tests. Molecular genetic method now very widely used, the most specific and the sensitive method. The aim of this method is detection of specific part of nucleic acid of the presumptive causative agent. And one test that is used, it is PCR. Now all mankind knows, all people know this word PCR, and it means polymerase chain reaction. But it is just one of reactions, maybe reverse PCR, maybe PCR in real time, maybe DNA zoned and other. The name of method is molecular genetical, molecular biological, and the last one, this method, skin allergic method. For this test, there are no laboratories. This test is done exactly in a patient's body. If you remember Mantoux test with tuberculin, it is done for children for diagnosis of tuberculosis and for in adults for diagnosis. Some microbes are able to cause allergic reactions in human body. Not all of them, some species of bacteria, some protozoa, most of fungi and no viruses. And commercial preparations, industrial preparations are produced that content of some substances of 13 microbes, like protein of tuberculobacteria, like spores of Bacillus anthracis. They are named tuberculin, anthraxine, lipramine, toxoplasmine, and so on. And this preparation is injected into the skin of patient for clinical diagnosis, and it is cell-mediated reaction, and we will see result in 3 days because of it is delayed type of hypersensitivity, not immediately. Every specimen should be sent to the laboratory, the specialized laboratory with presumptive diagnosis. And the aim for microbiologists is to detect these specific microbes, or maybe detect antibodies, and in between other microbes. Specimen from the mouth contain a lot of microbes. Specimen from the pus, from the wound, again, there are a lot of microbes, and feces and so on. And the task is to find the one species of microbes. Like we are looking for Vibrio cholera in case of cholera, we are looking for Bordetella pertussis in case of whooping cough. So we should know properties of microbes. And properties of species of microbes are every species of microbes has own properties, such as morphology, staining, cultivation, biochemical properties, pathogenicity factors, antigenic structure, and allergenic properties. And in every textbook, characteristic of every species is written by this scheme, all properties. And during 3 months, February, March, and April, you will study properties of microbes, and then after that, you will study separately causative agents. Now I will, doesn't matter, species of bacteria, species of viruses, species of fungi, or species of protozoa. They have the similar properties. And it's very important to learn by heart all these properties, because then you will study these causative agents, and hopefully you will know all their properties. So pay attention for general microbiology. This is a basement for medical microbiology. All you have to do, you should learn by heart and memorize. Now I will make a break, 10 minutes break, and then you have to join the lecture again by using all this link. Okay, I've sent the link. Leaders, do you hear me?
Yes.
Yeah.
So use the same link. In 10 minutes, we will continue the lecture.
Okay,
Okay.
doctor.
Okay.
Mhm.